Hello everyone, welcome back to the third week of the MOOCs course on Advanced Analytical Technique. Today I am going to start the second lecture of this third week where I am going to discuss about the instrumentation of the inductively coupled plasma technique. In the previous lecture, we have discussed about this particular figure where we have shown different components of the ICP in the form of a flow sheet. And since the very first component is the nebulizer, which is basically based on the different types of pumps which we use. So the very first component is the nebulizer, which is used as a sample delivery system. So let us discuss about this nebulizer. The nebulizer basically consists of a peristaltic pump that automatically draws the sample depending upon the previously adjusted flow rate. It sucks the sample from the sample container or the beaker containing this sample with the help of an inlet pipe, and then it draws the sample and it injects into the nebulizer. Now when the sample enters the nebulizer, we also have argon gas coming into this nebulizer, which converts the sample into an aerosol. And I hope you remember about the nebulizers that we have discussed in the atomic emission spectroscopy. We have the three commonly used nebulizers: the ultrasonic nebulizers, the nebulizers which works on the principle of Venturi called as jet or pneumatic nebulizers, and then we have a third type of nebulizer called as the electrothermal nebulizer, which first of all vaporizes the sample and then mix it with the gas to convert it into an aerosol. So any of these nebulizers can be used, and then we have one more nebulizer called as the Frit nebulizers that we'll discuss today. They uses a glass membrane to convert sample into small droplets that can be further converted into aerosol with the help of argon gas. So the peristaltic pump sucks the sample and pushed it into the nebulizer, where it is converted into fine aerosol with the help of our inert gas argon, with the help of our inert gas, and then it is being sprayed into the atomizer or into the excitation source. So in the spray chamber, the large droplets which are still not converted into very small droplets, they are settled down, while small droplets that have been evenly converted into aerosol, they are basically sprayed into the excitation source. Any excess liquid, any excess solvent is drained out from this nebulizer. So this is a schematic diagram showing this is our sample. Then we have this peristaltic pump. Then we have this peristaltic pump which pushes the sample into the nebulizer, and in this nebulizer the excess sample is drained out from towards the bottom, and then we have argon gas which converts this sample into aerosol, and from this argon gas based aerosol is then injected into the plasma torch with the help of spray chambers. This spray chamber sprayed the aerosol into the innermost tube of the quartz base plasma torch, where the sample is heated at very high temperature so that it immediately atomizes as well as excited, so that where the sample immediately is immediately atomized as well as excited to create an emission spectrum. So this is a schematic diagram. Then finally the emitted light radiations falls on the detector, and finally we can have sometimes sensor is mentioned because the detector is basically a sensor, and then we can have it on the computer. As I have already discussed, that nebulizer is the component which basically used to convert the sample into an aerosol. That can be done either by using air or oxygen gas, but in order to avoid the possibility of oxidation of the sample, oxidation of the elements that are present in the sample, in case of inductively coupled plasma, because the temperature is very high, the chances of oxidation are quite high. Therefore we avoid using oxygen or air. Instead we use an inert gas argon to create this aerosol. The size of the particles ranges from 1 mm to 2 mm or even lesser. Then the samples are injected into the plasma torch in its innermost tube with the help of a spray chamber. Then there are common type of nebulizers. These are pneumatic type. They are also known as jet nebulizers. And then there is another type which we have not discussed yet, the Frit type nebulizers. In addition to these, we have ultrasonic nebulizers as well as the electrothermal nebulizers. The pneumatic nebulizers we have already discussed. They works on the Venturi principle, or I should say the Bernoulli principle, in which you know that if liquid is flowing, or if gas, in which if a fluid is flowing through a pipe, and if the diameter of the pipe is reduced, the velocity increases while the pressure falls. On the basis of this concept, these nebulizers create a negative pressure or very low pressure outside the tube, so that the high pressure inside the tube pushes the aerosol with very high force, and it immediately enters into the excitation source. Then there are two types of nebulizer, concentric and cross flow. The difference between concentric and cross flow is that in this particular case we have the sample as well as argon gas. They are entered into this particular pipe, and they are at very high pressure. They have been, and they are being injected into this spray chamber at very high pressure, so that they are converted into aerosol. Here they are basically, here they directly entered into this pipe, they meet here and they start forming the aerosol. While in this particular case where we have a counter current, where we have a cross flow, this is called as concentric flow, because both the argon gas as well as the sample, they are being injected into the same tube. While in this case, argon gas is injected directly from this point, while the sample comes at 90-degree, called as cross flow, and then they meet into the spray chamber and form aerosol. This is the only difference between these two. Otherwise the principle is same. Then the ultrasonic nebulizer, we have a piezoelectric crystal which receive power from a direct current source and start vibrating at high frequency, creating an impact which causes the droplets to break down into small droplets, and then we have argon gas coming which convert these small droplets to aerosol, which is then injected into the plasma source. Any excess liquid is drained out from the bottom. This is a third type of nebulizers which we have not discussed previously when we were discussing the types of nebulizers in atomic emission spectroscopy. These nebulizers are basically, these nebulizers basically works on the principle of Frit membrane. Frit membranes are membranes which are, Frit membranes are glass based membranes. They are glass membranes, and they are quite porous as well. So through these pores, when the sample is passed, the sample is converted from large droplets. The sample forms very small droplets when it is passed through this Frit membrane, and then it is mixed with the argon gas. Here we inject the sample from the bottom, and the sample is pushes through this membrane to form these large droplets of the sample. They forms very small droplets, and then we have argon gas coming from the top, so we can say it is a type of counter current flow. Earlier we have seen a concentric flow in case of pneumatic nebulizers. We have also seen a cross flow. Now we can see the flows from the opposite direction. It is called as counter current flow, where gas is coming from the top while sample is coming from the bottom. They both mix with each other, forming a very super fine aerosol, and then this aerosol is passed into the plasma chamber. This advantage of Frit nebulizer is that they produce a very fine and uniform aerosol, and it is very ideal for efficient transport of sample into the plasma, or even for flame direct aspiration we can also use these Frit nebulizers. Then comes the IC source, the inductively coupled plasma source. We have already discussed it in detail. Just to give you an idea in the nutshell, it is basically a plasma source. In this case is called as a plasma torch, which consists of three concentric tubes, and in all these tubes we passes argon gas. In the innermost tube, the aerosol coming from the spray chamber which is connected to the nebulizer, this aerosol enters into the innermost tube. This aerosol contains argon gas plus sample, this one auxiliary argon gas at the rate of 10 to 20 liters per minute is injected to create the plasma, and in the outermost tube argon gas is used as a coolant to control the temperature and to protect this tube as well as the copper coils placed here for passing radio frequency are being protected with the help of this coolant argon gas. Here we have, as I said, copper coils which are connected to the radio frequency source. Radio frequency is passed, which has an oscillating magnetic field which creates very high temperature. Basically, that the mechanism is like that. Oscillating magnetic field creates electric current. As you all know that magnetic field creates electric field, while electric field creates a magnetic field. These two forces are not different, but they are one forces, and therefore they are called as electromagnetic forces. Earlier it was believed that they are only there are five forces, fundamental forces in nature: the gravity, the weak nuclear force, the strong nuclear force, electric force, and magnetic force. Later it was realized that electric and magnetic force, they are not different, but they are same thing, because when electric force is created, it automatically creates magnetic field, and the vice versa is also true. So they have been combined, and now there are only four forces: the electromagnetic force, the weak nuclear force, strong nuclear force, and the gravity. So this oscillating magnetic field creates eddy current. Those eddy currents creates very high temperature, and that high temperature causes this auxiliary plasma gas, and that very high temperature causes this auxiliary argon gas to ionize, forming Ar ions along with electrons. Although in the plasma there are some unionized Ar gas molecules as well, and the rate of the coolant argon is 13 to 20 liters per minute, while in case of auxiliary argon it is around 10 to 20 liters per minute. So we can say that in case of inductively coupled plasma, the requirement of argon gas is quite high, which is maintained by, I hope you know the answer, the Dewar flask, which is used to store all the liquids which have very low liquid temperature. Means argon gas which is having a temperature of -185, for nitrogen -196, and for helium it is around 260 degrees Celsius. They can be used, and that Dewar flask consists of two concentric tubes, cylindrical tubes made of stainless steel with a vacuum created in between to avoid heat losses through conduction or convection, to avoid passage of heat due to conduction and convection. And finally, this outermost cylindrical stainless steel uh, container is coated with silver to avoid any absorption of heat, to reflect all the heat. So the Dewar flask is used, and we have a plasma created over here. The plasma is very bright. It emits a continuum of bright light, but as we moves away from the plasma, as we moves away from this torch, the brightness of the plasma decreases, and at around 3 centimeter from this tube where the plasma is created, the temperature, the brightness goes completely, the brightness is completely lost, although we have very high temperature over there. So in this particular transparent region we ionize, so in this particular transparent region we excite the sample in the transparent region. Let us move forward. So again, it is written which I have already explained: the torch surrounded by high energy induction coil through copper wires, through copper coils, and radio frequency is passed, which creates eddy currents, which causes heat, excessive heat, ultimately converting argon gas into a plasma. And everything is being done in quartz tube, concentric quartz tube. The sample is introduced in the argon stream in the form of mostly liquid aerosol or vapor, but most of the time we prefer liquid aerosol, and it enters from the innermost tube, and at a distance of 3 centimeter where the zone is transparent, we do the analysis by creating an emission spectrum. So this is all about, these are three concentric tube. This is the innermost tube. Then we have this opening for passing auxiliary gas, which creates a plasma, and this is for coolant gas inlet, which is again argon. Liquid argon is used to control the temperature and protect these protect the whole assembly. So we carry out the analysis where we have a transparent region. Here we have very bright continuum, so we do not analyze or excite the sample. Here we analyze the sample at a distance of around 3 centimeter, so that we can have no interference or minimum interference. This plasma has different temperature ranges. Even we have 10,000 degree Kelvin temperature range, that can also be possible. It is at the center of this plasma, but as we move away from the center, the temperature decreases. At immediately at the center, when the distance is 0, we have 10,000 Kelvin. At 115 millimeter means 1.5 centimeter, the temperature is reduced to 7,000, but still it is very high temperature. And at around 2.5 to 3 centimeter or 25 to 30 millimeter, the temperature is higher than 6,000 Kelvin. But this particular region is transparent region, and therefore we carry out the analysis and excite our sample to create emission spectrum in this transparent region only, just to avoid any background radiation. Otherwise we would be having large number of otherwise we would be having excessive of background radiation, which makes the analysis highly complicated. Then again we have monochromators. As I said earlier, monochromators are very useful device. They are used to select particular wavelengths, and they are also helpful to exclude the other non-desired wavelengths. Although in case of atomic absorption spectroscopy, we use the monochromator before the light path interact with the sample. Atomic absorption, where the light radiation is coming from this light source. Light radiation is coming. So this is the light source where the light radiation is coming, and then we have an atomizer which is connected to a nebulizer. So this is our nebulizer. We have atomizer over here. Here the sample is injected from the nebulizer and converted into aerosol, where it interacts with light. So before this atomizer, we use a monochromator over here, so that we can select a particular wavelength of light in which we want to make the study. While in case of atomic emission spectroscopy, because no light source is used initially, the light radiations is created by the sample itself. Therefore we use, first of all, we have a nebulizer which is connected to an atomizer or the excitation source. Now here in this particular case, this is our excitation source of atomic emission spectroscopy, or more specifically I should say the inductively coupled plasma. For example, if this is our plasma torch, and sample upon heating and excitation, and the sample upon heating and excitation produces emission spectrum. So light is coming from here. So we use a monochromator after the excitation source. This is the monochromator. So always remember, in atomic absorption spectroscopy, we use monochromator before the atomizer is placed. Just after the light source, we use monochromator, we select light, and then the selected light, monochromatic light, enters into the atomizer for absorption. While in case of atomic emission spectroscopy, we do not use that. We use monochromator after the excitation or atomization source. Again, monochromator, as you all know, consists of a grating or a prism. So we can use prism, or grating can be used to disperse light, distribute light into different wavelengths. Small, they basically monochromators basically narrow down the wavelengths. For example, if light radiations is coming from the wavelength from 200 to 400, then monochromator can be used to convert this light radiation with a wavelength of 200 nanometers. From 200 to 400, it covers a region of 200. It converts it into, say, 50 nanometer. And if we want to have more specific wavelength, we may use prism and grating together, so that we can have, first of all, a 200 nanometer wide light radiation is converted into 50 nanometer, and then prism or grating is used again, which converts this 50 nanometer again into 10 nanometer. So we can bring down the wide gap of this electromagnetic radiation of 200 to, and make it very narrow. We bring it down to 10 nanometer. So for this purpose, monochromators are used. So these monochromators are very useful because they reduces spectral interferences. If we are not using monochromator, there are chances of having spectral interferences which may spoil the analysis. Therefore monochromators are always very useful, because they basically act as a filtering device which filter some of the desired spectral lines only while cut off the other. And both prism or grating can be used. Most of the cases we prefer grating. In some cases where we need to narrow down the light up to a greater extent, we use grating as well as prism both. Sometimes holographic grating is also used to create light radiation into individual components. With this, we have completed the components of inductively coupled plasma, like the nebulizer, the excitation source, and monochromator. In the next lecture, we will discuss about the two common types of spectrophotometers that are basically preferred for atomic emission spectroscopy. In one case, the first are called as sequential or slew scan spectrometers, while in second case we have simultaneous spectrometer that are used for the detection of multiple element at a time. So we'll discuss that in our next lecture. Till then, thank you very much.